Good morning, student. Today we are discussing about the cyclic and chain structure of carbohydrate. As we know, carbohydrate present in chain form, linear form, or also it is present in cyclic form. We know that the cyclic form of sugar or saccharide or carbohydrate represent in two forms, which are which are pyranose and furanose. This name shows pyranose and furanose. The name shows pyranose means there is a six member present in a ring, and furanose means it has five member in a ring, but there is a furon-like structure. In a cyclic structure, in linear structure of carbohydrate, the one of the hydroxy group from five number or four number carbon of carbohydrate, it react with the aldehydic group or ketonic group, and it convert into the hemiacetal conditions or hemiacetal functional group, and it form the ring. So it form this ring, and if there is a six member present in this ring, so it is a pyranose type of ring. And if there is a five member present in the ring, so it is known as the furanose type of ring. So in two class we can classify it again carbohydrate. This is exactly happen in glucose. In alcohol means hydroxy group of say fifth five number of carbon. In glucose, it react with the aldehydic group on a first carbon atom and form the six-member ring. So it is called as a pyranose-like structure. I skipped drawing in a proton transfer in drawing below. The linear and cyclic form are structural isomers that exist in equilibrium with each other. So this is another example of ring chain tautomerism. The six-membered cyclic form of sugar is usually called the pyranose form in reference to the cyclic ether pyrene. Means after the forming the sixth member of the ring, the structure is like a pyram, but it is a sugar, it is a carbohydrate, so their name is nose. So that is why it is a pyranose structure. If you are eagle-eyed, eagle-eyed, you might have noticed that in process of forming a new CO bond, a new chiral center is formed at C1. This new chiral center can have one of two configuration, S or R configuration. Since there are another chiral center on glucose and their RS configuration don't change, that means we will end up with a pair of distereoisomer and stereoisomer that are not in enantiomer. Rather than using the RNS descriptor, the convention with sugar is to name them according to the orientation of the OH group on a C1 carbon in a glucose molecule relative to the C5 group. These two isomers are referred to as the alpha and beta isomer. As we know, at the first carbon in glucose and in ketose, there is a aldehydic group and ketonic group, and from the fifth number of carbon, if the or four number of carbon, when in pyranose there is a five number of carbon hydroxy group react with the first number of aldehydic group and form the cycle. But there is a new stereogenic center is formed in a cyclic pyranose structure of the glucose, and their configuration may be R type or S type of configuration, but the configuration at the another carbon atom in a glucose as it is present in a cyclic structure. But after the reaction of hydroxy group with the first carbon atom of the aldehyde in pyranose structure, their position of this new hydroxy group in a pyranose structure decide the name of or stereoisomers or their isomers of the pyranose. whether they are alpha type or whether they are beta type, that depend upon their position of newly forming stereogenic center. The straight chain form of glucose is equilibrium with the cyclic six-member ring form in a glucose, the pyranose form. So there is a chain form and cyclic form equilibrium is obtained. Here we can be seeing at the fifth number of hydroxy group. This hydroxy group interact with the first number of carbonyl group of the aldehyde, and this center, this C double bond O, is converted into the C single bond O, and there again there are four different type of substituent on this carbon. That's why this carbon again become a chiral center, and it may be the alpha type or beta type, that depend upon their position of this hydroxy group, but there is a equilibrium in between to the chair form and cyclic form of pyranose. Six-member ring. Here the six-member ring is formed, that is the first carbon, the second, the third, fourth, fifth, and this oxygen is a sixth number. So this type of structure is formed here. So this is called as a pyrene structure, and here also form this glucose molecule also form this type of structure, so it is called as a pyranose structure. And at this first carbon atom there form the new stereo center is formed. The six-member ring form of sugar is called as pyranose form in reference to the cyclic ether pyrene. Interconversion between an open and closed form of molecule is called a ring chain. So this is the ring chain tautomerism. This is a chair form, it convert into the ring form. It just because of this attack of hydroxy group on this carbonyl group. That's why this is called as, there is a conversion of this carbonyl group convert into the hydroxy group. This conversion is known as a tautomerism in between to the chair form and cyclic form. In alpha isomer, the hydroxy group on the first carbon atom on the opposite face of the ring from the CH2OH substituent on carbon. This can be seen from drawing the molecule as a chair, but it is often helpful to draw the hexagonal version of the sugar particle called Howard projection. This is the Howard projection representation that make the stereochemical relationship more clear. In the beta isomer, the hydroxy group of first carbon on the same face of the ring relative to the CH2OH substituent on C fifth carbon atom. Means relatively the position of hydroxy group form at the first carbon and hydroxy group on the C fifth carbon, so that decide relatively the position of fifth hydroxy group and first carbon hydroxy group, their isomers are, names of the isomers are given, that is alpha and beta. In case of alpha, here we are seeing here this hydroxy group is present above the plane and of the fifth carbon atom, that is CH2OH, and here the hydroxy group of the first carbon present below the plane, so their name is called as alpha D glucose. And here we are seeing the position of hydroxy group on the fifth carbon and hydroxy group on the first carbon, both are present on the same phase, then it is called as a beta D glucose. And here hydroxy group of fifth carbon and hydroxy group of first carbon present on opposite side, so this is the alpha D glucose. The two stereoisomers of glucose in its pyranose form are called the alpha and beta isomer, which differ in their orientation. Furanose structure. It also possible for the hydroxyl group of C4 number of carbon attack on the aldehydic or ketonic group on the first carbon and from the ring, but this ring is contain the 5 member and this ring like an furon like a ring. That's why their name is known as a furanose structure. As with the pyranose, forming the 5 member ring has generated a pair of distereoisomer which differ in configuration at C1. We likewise call this alpha and beta form. And similar to the pyranose, in a furanose also, the position of newly hydroxy group form on the first carbon in a furanose, their position relatively in relative to the position of relatively the position of hydroxy group on the fourth carbon atom that decide the name of isomers formed during this chain formation. Glucose can also adopt a 5 member furanose form in addition to 6 member pyranose form. Here, glucose molecule which contains the 6 carbon atom and 6 hydroxy group, 5 hydroxy group and 1 carbonyl group, aldose group. So here, fourth number of carbon which having this hydroxy group. This hydroxy group react with carbonyl group and form the ring. And here this ring is formed. In this ring there is a 5 member is present. This first, second, third, fourth and this fifth one is oxygen. So after forming this ring like the ring like furan, and it is a glucose molecule, so their name is furanose. At the first carbon, due to the attack of this hydroxy group, this new stereogenic center is formed because it contains the 4 different type of substituent. This ring is formed is a 5 member ring and new stereo center form at first one carbon first. Glucose also has alpha and beta isomer in a furanose form which are assigned in similar fashion to the pyranose form. Means the position of this hydroxy group is first carbon hydroxy group relatively to the position of fourth carbon hydroxy group. If both are present on the same side, so it is called as a beta glucose, and when it is present on opposite side, then it is called as a alpha D glucose. So here this fourth carbon hydroxy group present on this side but exact opposite hydroxy group present on the first carbon, so it is called as alpha D glucose. Here on the fourth carbon the hydroxy group present on upper side and on the same side the hydroxy group on the first carbon atom in furanose is present, so this is the same phase of the ring, so this is called as the beta D glucose. Glucose has several structure all in equilibrium with each other. We have seen 5 separate isomers so far. The straight chain form the pyranose form alpha and beta and furanose form alpha and beta. In aqueous solution, these 5 form are all in equilibrium with each other. When you dissolve glucose in water, here the distribution you get. As we know, the larger ring become more stable. So comparative to the pyranose and furanose, there is a pyranose ring is a 6 member ring, so it is a less steric ring. That's why it become a more stable as compared to the furanose, because furanose ring is smaller as compared to the pyranose ring. Second one, this it gives one chain form, another is cyclic form, but cyclic are 2 type. One is pyranose and second one is a furanose. Pyranose again gives 2 type of isomer, alpha and beta, and again this furanose also gives 2 isomer, alpha and beta furanose. So in water when dissolve this glucose, it gives a 5 type of structure, and these all are present in equilibrium. But how if you seeing this out of this, so there is a six member ring. This pyranose form of the glucose is become more stable as compared to the chain form and furanose form. That's why in the water, in the solution, there is a 99% of pyranose form, because pyranose form dominate, which small amount of open chain and furanose form comprise the rest of the mixture.